we're going to try again to do that GFP lab. So take a look at that, and your RNA to protein ratio lab report is due. Okay, also a reminder, if you're considering dropping this course, you have until Friday to do that. Friday's the last day you can drop courses for the semester. Okay, we've been talking about the endoplasmic reticulum, mostly the rough ER so far. We'll get to the smooth ER shortly and get into the Golgi apparatus today as well. And we'll do the lysosomes on Wednesday. Okay, so we saw that this whole endomembrane system, the endoplasmic reticulum, the Golgi, and lysosomes are sort of designed for three broad categories of proteins. Those that are going to get secreted from the cell, those that are lysosomal enzymes, so will end up in lysosomes, and those that get inserted into the membrane. Okay, so those are the three types that we're going to focus on, that we've been focusing on in this section. All the other proteins of the cell, those that stay in the cytoplasm or go to other organelles like the nucleus, mitochondria, etc., are made on ribosomes that are just sort of floating around in the cytoplasm. Okay, so the first step that we saw was that we have to get those three categories of protein to the rough ER. And we saw that to do that, we needed a signal. That signal was the first 16 to 20 amino acids. If that whole stretch is hydrophobic, that is the signal sequence to get that protein to the rough ER. And we saw that this little particle here, the signal recognition particle, proteins and RNA molecules, have the job to recognize that signal sequence, bind to it, that stops translation, and eventually that whole thing will bump into the rough ER where there is a receptor for that signal recognition particle, and those two dock together, and that puts the ribosome on top of this translocation complex. And the little lid opens if you remember that animation. The SRP threads the protein into the translocation complex. It leaves to do this all over again, presumably. Translation, and the protein is continually translated and pushed through this translocation complex until it gets to the inside of the ER. So this was our example for either a protein that's going to get secreted, or this could be a lysosomal enzyme. And we saw it needs to get inserted into the membrane. We have, in this example, a second signal sequence, which is kind of just like the first one. It's this stretch of hydrophobic amino acids, but in this case, in this context, we call it the stop transfer sequence. Again, it stops translation and then somehow interacts with the translocation complex to be inserted in the membrane. So this is how we get proteins inserted into the membrane. Okay, and it will stay inserted through the rest of its journey, as we'll see. And then we started looking at modifications that can happen proteins while they're in the ER. So we talked about picking up sugars. So there was yet another signal for that. Asparagine, X, threonine, or serine. If you have that 3 amino acid signal, you pick up this sort of prefabricated set of sugars that can be further modified. We don't completely understand the signals for that part, but we do understand it to get this whole sort of chunk of sugars in the first place. So that's called glycosylation. And I think that's where we stopped. Is that right? Okay, so how are we feeling about getting these types of proteins to the ER, inserting proteins into the ER membrane and picking up sugars? Okay. Let's hold up on that one. A moment. Okay, so our first post-translational modification then was glycosylation. Our second one is part of the whole folding process. Okay, so remember, proteins, once they're made, start folding right into secondary and tertiary structure at least. Well, one enzyme that kind of helps this along inside the ER is essentially an enzyme in a sense called protein disulfide isomerase. This protein has a lot to do with forming correct disulfide bonds. And remember all the way back to, I guess it was September when we were doing this, and if we had two cysteines close by, the ones with the SH on the end of the R group, those can form a covalent bond, right? We call that a disulfide bond. Well, that's great, but what if we had a situation sort of like this? And there are 4 cysteines in this protein. So we can make 2 disulfide bonds. But the question is, which 2 go together? So is it 1 and 2 go together and 3 and 4? Or is it 1 and 3 and 2 and 4? Or is it 1 and 4 and 2 and 3? Right? Only one of those combinations should be the correct one. Well, as this protein is folding, let's say it forms those 2. Well, what if that's wrong? What if that's not the correct set? What would happen to this protein if it made incorrect or formed incorrect disulfide bonds? Yeah, it's not going to fold properly, so it's not going to have its right shape. If it doesn't have its right shape, then it's not going to have its right function. So what this protein is able to do is, as the name suggests, it can isomerize disulfide bonds, which basically means it has the ability to sort of kind of know, quotation marks, that this protein hasn't folded correctly, and it has the ability to break those disulfide bonds and sort of give the protein another chance. Because otherwise, the cell has gone through this very long, very complicated, very energetically expensive process to make this protein, and if it just makes that one mistake, then it's dead. And the cell has wasted all that time and energy. So this gives it another try, and let's pretend this time, oh, I don't know, it does 2 and 3 and 1 and 4. And let's say that's wrong too. Well, again, protein disulfide isomerase has the ability to say, okay, that wasn't right either, let's give it another try. So we can break those bonds, give it another try, and let's pretend this time which combination did I do? 1 and 3, 2 and 4. And let's say that's correct. Somehow protein disulfide isomerase knows that that's right, possibly because maybe when it's correct, those disulfide bonds are buried or something like that. We don't really understand exactly how it knows when to break them and when not to. But it does give it, in short, multiple chances to form the correct combination of disulfide bonds. So this is a protein that's inside the lumen of the ER. So it's floating around in here, and like I said, gives it a chance to fold correctly. That kind of makes sense? So that's kind of about folding. And then, continuing on the folding idea, most of the time proteins can do all their folding on their own. Now again, sometimes they might need a little bit of help from PDI in terms of making sure their disulfide bonds are correct. But some proteins need a lot more help in order to fold correctly. And that help is given by a large complex of proteins that are called chaperone proteins. And the long and the short of this, and we're going to do the short version, is that these proteins help other proteins to fold correctly. And we could spend a week talking about chaperone proteins, if not a whole semester, so we're just going to look at sort of this definitionally. So there are, sort of drawn here in red here, other proteins that collectively are called these chaperone proteins and help other proteins to fold correctly into their correct tertiary structure. So some proteins need that help, many, probably to most, do not, but there is this sort of additional help for proteins to fold correctly. Again, for the same reason. If this stays misfolded, then it's not going to have its correct function. Okay, so again, the cell has wasted time and, more importantly, energy making that protein that didn't fold correctly. If we just give it a little bit of help, and we get it to fold correctly, and we get another functional copy of that protein. So things that are sort of happening to some proteins inside the ER are mostly sugar-based, right? We pick up some sugars, and kind of these two are both kind of about correct folding of the protein inside the rough ER. Either through the help of protein disulfide isomerase and or these chaperone types of proteins. So hopefully all the stuff that's going through the ER is going to fold properly, and the ones that need sugars will pick those up as well. Yeah.
So the chaperone protein is, the PDI right there is not an example of the chaperone.
It is not. This is a separate thing just for that, just for this idea for disulfide bonds. This is a much broader idea of overall folding. Any questions? Okay. I got a picture, okay. Let's go back to this picture then. Okay, so that's sort of the basic story with the rough ER, so all about proteins. And we'll come back to this and move from the rough ER to the Golgi, or move those proteins from the rough ER to the Golgi. But I just want to take a little step aside and talk just for a couple minutes about the smooth ER, which we haven't really talked anything about yet. So we have this completely distinct type of endoplasmic reticulum called the smooth ER. And as we talked about last week, and we can see in this picture, the morphology changes, right? The shape of this part of the ER changes, even though they're connected. Okay, so it's one contiguous organelle, but you have sort of a rough section and a smooth section here. And as we talked about briefly last week, if the shape changes, probably the function is going to change, and that's definitely true. Okay, so smooth ER having a more tubular shape and no ribosomes attached has really very, very little to do with anything about protein. What the smooth ER is going to have a lot to do with, really two things. First is lipid production. So instead of dealing with making certain kinds of proteins, the smooth ER is much more about making lipids of various kinds, but most notably, this is where a lot of the new membrane phospholipids are made. They are going to be in the smooth ER, and then they're going to travel eventually to the cell membrane. This is also the site of cholesterol synthesis, so lipids in general are made here in the smooth ER, so it's much more about lipids rather than proteins. And then the other big thing that the smooth ER is all about, in all cell types, but especially in the liver, is the predominant place where we see detoxification. So if you look at liver cells, they have a lot more smooth ER than your average, say, skin cell or brain cell, or pick any of your other favorite cell types. Okay, so there's a lot more smooth ER because, as you're probably aware, your liver is our main sort of detoxifying organ. So there are inside the smooth ER a whole family of enzymes called the P450 cytochrome family of enzymes, and they are all about detoxifying the nasty things that we eat and or drink. So, if anybody was out drinking this weekend, it was your smooth ER that was essentially sobering you up, right, by detoxifying the toxic alcohol that you were imbibing this past weekend. So it does that. The major thing that these enzymes do is add mostly hydroxyl groups to other compounds, which makes them more soluble and therefore more easily excretable from your system. So it serves usually a very, very nice function. However, there are some instances where you don't necessarily, or you would prefer them not to do what they do. The one big example of that is in the pharmaceutical industry. So if you're working for Merck or AstraZeneca or any of the others that still exist, not merging with each other, this is one of the first tests that you run if you think you have a new, blockbuster drug that's going to do whatever, kill cancer cells or lower somebody's cholesterol or stop people from having a heart attack, whatever. You want to know what these liver enzymes are going to do to your drug. Because sometimes what happens in a test tube outside the body and what happens inside the body are very, very different. And that's because of these enzymes. So you might have a great new potential drug in a test tube, but once these enzymes get a hold of it, they don't work anymore. So adding hydroxyl groups can inactivate compounds. So sometimes something that looks like it's going to make you $1 billion in a test tube, these enzymes get a hold of it, and it's not worth anything when it's actually inside your body. So you want to know that kind of early on before the company has invested, $100 million in it, only to find out, oh, right, when you actually ingest the pill, it doesn't work anymore. Right? You want to know that sooner rather than later. So this is one of the first tests that you run if you're working for a pharmaceutical company before they invest a lot of money in your project. So that's one thing. And the other big thing is sometimes they actually do more harm than good. Sometimes you can ingest something that's not going to hurt you at all, but then these enzymes get a hold of it, add some hydroxyl groups, and actually make the compound harmful. So luckily that doesn't happen very often, but it does happen occasionally. So as most things in biology, there's mostly a good side, but there's always a little bad side to the idea. So sometimes this can be kind of the opposite of what it was really designed to do, but like I said, luckily that's kind of rare. Most of the time these do exactly what they're supposed to do and detoxify things so we can excrete them fairly easily. So that's 2 minutes on the smooth ER. Well, I guess one other kind of example, actually I should mention, in muscle cells, the smooth ER is given a different name. It's called the sarcoplasmic reticulum. And this has a lot to do with controlling calcium levels in muscle cells. That's another thing, that's another good example. Alright, that's probably plenty about the smooth ER. Okay, so lipid metabolism in all cells, detoxification, well, technically in all cells, but very much so in the liver, and then we have this calcium control in muscle cells predominantly. Although this is not exclusively the calcium level, just in muscle cells, but it's a really good example in that particular cell type. Yes?
Isn't increasing calcium related to secreting a bunch of other stuff within the cell?
Right. That's why I just said what I said. So this is not just about muscle cells, but.
And the cytochrome, didn't we study it before in photosynthetic plants, and they move the electrons from one to another?
They did.
Is that basically how they attach the hydroxyl group to it, or?
I
just leave it?
I don't think so. I don't think mechanistically that's the same.
Are they related?
I would think so, if only because of the name, but the extent of that homology, I don't know. Questions about the smooth ER? Okay, so let's get back to the rough. And now we're ready to move those proteins that we have added some sugars to, that we have helped fold, perhaps. The next destination for the majority of those proteins will be the Golgi apparatus, okay, the second of the three pieces of the endomembrane system. So, how do we get proteins from the rough ER to the Golgi? By packaging them in little vesicles that bud off the ER and then move to the Golgi and merge with the Golgi membrane. Okay, so we've kind of seen this before. Think back again to probably around September when we talked about membrane-assisted transport and secretion and things like that. We saw that membrane-bound vesicles can fuse with other membranes. Again, think about your little blue soap bubbles. Two bubbles can fuse together, right? It's the same kind of idea. So that sounds like a very simple idea, except how did the vesicles know where to go? Why don't the vesicles move from the ER directly to the cell membrane or fuse with any other kind of thing inside the cell? How come they go from the ER to the Golgi? Well, at least part of this story has been worked out, and the part that we're going to talk about are special proteins called snares. So apparently the answer to this question is that there are specific sort of locator proteins in these vesicles that can team up with the sort of second piece to the puzzle only on specific organelles. So there are two kinds of snares. And you need kind of one of each to get where you're going. So there are first V snares, V for vesicle. So there's a snare protein in the vesicle. So in this example here, the V snares would be the red one here on this vesicle, and the sort of brownish one or tannish one here on that vesicle. And then on wherever they're going, the target, there is what's called a T snare for target snare. Okay, so for target organelle, so where is it going? And there are specific combinations, as they're trying to demonstrate here in this figure, sort of by color coding them. So I wish they would have color coded them a little bit better, but we'll deal. Okay, so the red V snare can only hook up with this sort of darker blue T snare, not the lighter blue. I wish they would have drawn that one like green or something, but you get the idea. Whereas this sort of tan one can only hook up with the light blue T snare and not the dark blue T snare. So that means this vesicle is only going to this compartment, whatever that is, and that vesicle is only going to compartment A. So it's a nice little system where only specific V snares can hook up with specific T snares, and that gets you where you're going, assuming all of this kind of works out. Right? You put the right V snare on the right vesicle with the right contents, and the right T snare is on the right target organelle, then you can direct specific vesicles with specific contents to specific locations in the cell. Okay, so most of the vesicles coming off of the rough ER have a V snare that hooks up with a T snare on the Golgi. Okay, so that's how we get them where they're going. Now this ends up causing us to ask a lot of additional questions for which we don't have the answers yet. How does this actually work? So how does the cell set up the system where the right cargo in the right vesicle has the right V snare to go here versus this one? That part we don't completely understand yet. Okay, but this seems to be sort of the general pattern. And what these snare proteins seem to do is to bring the vesicle membrane very, very close to the target membrane, the organelle membrane, so that they can then fuse together. So that's kind of what seems to happen. So we know at least part of the story involved these proteins called snares. Still a lot to work out. We're in the part of the course where I'm going to be saying that an awful lot, where we know part of the story, but we don't know the whole story quite yet. And so, again, if any of you are sitting here worried that we know everything there is to know about cell biology, I keep showing you that we don't. Okay, so some of you need to go to graduate school and figure this out. Because again, after 15 years of teaching this, I'm tired of saying we don't know this part yet. But we know more than we did. I've only been teaching the snares probably for 5 or 6 years. Okay, so we do keep learning things. So this is at least part of the story of how we get specific vesicles to specific places. So, like I said, the majority of those proteins coming off the rough ER are headed to the Golgi, okay, our second location. So all cells have at least one of these organelles. Certain cell types are going to have more than one. So your pancreatic cells, for example, that secrete insulin a lot will probably have more than one Golgi apparatus. Cells in your stomach that secrete mucus all the time will have lots of these. But every cell has at least one Golgi apparatus, Golgi stack. Goes by several names. But they're sort of a stack of little pancake sort of shaped structures that are called cisternae. I'm just going to draw them sort of simply here. You can see the shapes are more complex, but I'm just going to draw them kind of simply here. There's some good evidence that these things are sort of attached together. The stack sort of stays together, stays connected. But they're not contiguous. So it's not sort of like this, where it sort of looks like the things are separate, but they're really connected. Okay? Each cisterna is separate, but they're sort of kind of attached to the next one in the stack. And the way we sort of look at them, so the rough ER is up here, and the cell membrane is here. The way we usually refer to these different cisternae is that the ones closest to the rough ER is called the cis. The ones in the middle are called the medial. And the one either furthest away from the rough ER or the closest to the cell membrane, however you want to think about it, is called the trans. So we have cis, medial, and trans Golgi. And the only reason we really care about those designations is that different things happen to proteins in the different types of cisternae. So that means that there are different enzymes sort of sequestered in the cis versus the medial versus the trans. Okay, so again, the ones closest to the rough ER, so that must be over here, are the cis. The ones furthest away are the trans. The ones in the middle are medial. And we, as you can see in this image, we can stain specifically for say, the medial ones here, as opposed to the trans or the cis. So that must mean there are sort of biochemical differences between these three types of cisternae. And this kind of gives a much more detailed view of what we're going to look at next than I need us to worry about. So I'm not going to care too much, except with maybe one exception, of where things are going to happen. I'm just going to kind of summarize all of these into four major categories. Okay? So, and except for when we get to lysosomal enzymes, where we will care a little bit about cis versus trans, we're just going to kind of look at these in general. But we do have a fairly good idea of where different things are happening as a protein travels through this Golgi. But again, for our class here, I'm not going to worry about that removal of mannose happens in the third medial cisternae. I'm not going to worry about that. We don't have to worry about it quite at that level of detail. So I'm just going to basically summarize all these different bullet points into four sort of large categories. So the overall idea of the Golgi is twofold. One, it's going to continue to modify proteins. So we're going to continue with our post-translational modifications. And then the other big thing that's going to do, especially by the time the proteins get to the trans part of the Golgi, is to sort them to their final destinations. Okay, so Golgi is all about post-translational modifications of proteins and protein sorting. Okay, so we'll look at both of those functions right now. So we'll deal with the post-translational modifications first. And our first one is one we've already dealt with. and that is continued glycosylation. So proteins can continue, as they travel through different compartments, pick up more sugars. And we can see that, again, as starting here in the cis, they can pick up N-acetylgalactosamine. They can have phosphates removed here, mannose removed here, but attachment of N-acetylglucosamine, addition of galactose, addition of sialic acid. So it happens kind of throughout the Golgi, but in different compartments they can pick up or lose different kinds of sugars. But again, if you want just a general idea, more sugars are going to get added and sometimes subtracted from the proteins as they travel through. Oh, and I should mention, just so it's clear, since there's not a direct channel between the different cisternae, how do we get from the first one to the second one? Again, it's vesicular traffic. So a little vesicle will bud off of this one and then fuse with the second one, and then bud off from the second one and fuse with the third one, etc. So they sort of make their way through or down the Golgi stack, if we're looking at it sort of from top to bottom. And sometimes you will see them kind of going backwards as well. So that can happen too. So they can go backwards from, say, the fourth to the second one. So there's vesicles kind of moving all over the place in and around the ER and the Golgi. So if you look at that under the microscope, that's the area of the cell where there's just a ton of vesicles moving all over the place. Okay, so that one's kind of simple, I think. The second modification that can happen is adding sulfate groups. So it's called sulfation. So some proteins get sulfate groups added to them in addition to or instead of sugars. So that's the other thing to realize. Not every protein gets every one of these modifications as they go through. Some get some sugar and none of the others. Some get sulfate and sugar. So pretty much every combination is possible. So some things will pick up sulfate groups. A couple of good examples of this is a protein called mucin that's secreted by cells in your stomach called goblet cells. And mucin, as its name sort of suggests, is part of the mucus that coats your stomach and protects it from being destroyed by the stomach acid. So this particular mucin protein has a lot of sulfate groups attached to it, as it turns out. And another good example is a protein called chondroitin sulfate, which you might be familiar with. Sulfate, which is secreted by cartilage cells. And this helps sort of lubricate your joints. This is now a very popular supplement for people with joint issues and people with arthritis. And there are some good studies now that show that it does actually work. We're not exactly sure how, but it does seem to work, that if you take that supplement, it does seem to help a lot of people's joints. So that's no longer sort of, just them trying to sell you supplements. That does seem to be actually the case. That's right, that's exactly right. Oh, what I meant that we didn't know was how it gets from your stomach to your joints. That's the part we don't understand. Because you take it as a pill kind of idea. So why this isn't all getting broken down and how it gets from your stomach to your joints is the black box. Because there are some good controlled clinical studies that do show that it does work. But as its name suggests, so it's in the name, so this is also a highly sulfated protein. Just a couple of examples there. So that's our second thing. Our third thing, this is back here, is so we can pick up sugars, they can pick up sulfate groups, they can also pick up phosphate groups. So that will be our third modification, phosphorylation. phosphate groups. And then the final one is shown here is called proteolytic processing. Some proteins are initially made in a larger, usually inactive form. So insulin is the example that I have before you here. Insulin is initially made as a much longer protein. That's called pre-proinsulin, is the initial product that comes off the ribosome. And as pre-proinsulin moves through the Golgi, it gets proteolytically processed. Pieces of it get removed. And it's sort of, for insulin, it's a two-step process. So the first step is cleavage of the blue part here, the signal sequence. Okay, so I guess sort of technically we've already seen this. So when that first 16 to 20 hydrophobic amino acids get removed, that's technically, I guess, a post-transcriptional modification even back then, so that's proteolytic processing as well. So the signal sequence gets removed. So that actually happens back in the ER. In the Golgi, the red piece here gets removed as well, but interestingly, and the two green pieces stay stuck together because of disulfide bonds, which is kind of interesting. So they form first, and then if you cut out the red piece here, the two green pieces stay together, and that's the active form. So when you remove the blue piece, pre-proinsulin becomes proinsulin, and then when you remove the red piece, it becomes actually insulin, which actually works, which actually does what it's supposed to do, which is to signal other cells that you've just eaten a candy bar, and there's going to be a flood of sugar coming their way, and they should absorb that and use that. So insulin is a really good example of this proteolytic processing. Another good example is a protein called in. This is a protein that helps move fatty acids around your body. So they hook up on this protein called albumin, and that's how they circulate. But this is made in a larger form first, but a little less complicated here. So it's just it's just proalbumin gets proteolytically processed to albumin. That's another example. So this is, other than the pulling off of the signal sequence, which is very common, further proteolytic processing is relatively rare, but it does happen enough that it's one of our four big post-translational modifications in the Golgi. So this is kind of just a brief summary of all of these different bullet points. We can put all of those into one of these four categories. So proteins can pick up some sugar, will pick up or lose sugar, pick up sulfate groups, pick up phosphate groups, and or be proteolytically processed. But again, realize that not all proteins do all four of those things. Okay, some just get sugar, some just get sulfate, some get sugars and phosphate groups. Any combination really is sort of possible. Yeah.
Why didn't inside of the ER it didn't go through the one that we talked about PEI, where it just bonded the disulfide bond there?
Right. What about
disulfide? You said that one was happening inside of the Golgi, where it has the disulfide bonds. If you go back to the picture.
I said that there are disulfide bonds in insulin.
Oh, so they're already there.
Yeah. Yeah. This is what's entering the Golgi. This is what comes out. Processed version. Okay, so these are the four big things that can happen to proteins as they go through the Golgi. Before we get to the sort of sorting idea, one last sort of connection between the ER and the Golgi. While most of the proteins are sort of traveling sort of one direction. So they've been to the rough ER, they move to the Golgi, they move through the Golgi, etc. There are a few proteins, like protein disulfide isomerase, for example, like those chaperone proteins, that may need to go to the Golgi, get modified, but then where do they belong? Where do they work? In the ER. So some proteins, not a whole lot, but a few, have to get back to the ER from the Golgi. So hopefully we're getting used to this. Those proteins, of course, have to have a signal to do that. Very good. And the signal to do that we've worked out, and it's called the KDEL signal, or the KDEL sequence. KDEL is a 4 amino acid sequence. This is the single letter abbreviation for the amino acids. So K is lysine, D is aspartic acid, E is glutamic acid, and L is leucine. So if you have that 4 amino acid sequence right in a row, that's the KDEL sequence. And there is presumably a receptor that sees that, and if you have that, so in this example here, we have the blue protein, the green one, and this little sort of wishbone-shaped protein. Of the three, the only one that has the KDEL sequence is the little wishbone-shaped protein. So while the other two, the blue and the green, travel through the rest of the Golgi, the KDEL sequence says, or targets, this little wishbone-shaped protein to go back to the ER. Okay, so maybe that's, again, protein disulfide isomerase, or the chaperone protein, or something like that. Okay, if you don't have that sequence, if you don't have the KDEL sequence, then you are continuing through the Golgi. Okay. So you're working your way through the Golgi, you're picking up some of these modifications, and then you hit the trans-Golgi, and now it's time for you to be sorted. Okay, so the other big function of the Golgi after modifying proteins is to sort them to their final destination. So by and large, most of these proteins are going to make their way to the cell membrane. Okay, so if you're a membrane-bound protein there, okay, so if you're stuck in the membrane, then you're going to travel through a vesicle again to the membrane. When that fuses with the membrane, this one's a little better. Okay, so there's a little black protein here. Okay, when this vesicle fuses with the membrane, then that protein is now in the cell membrane. Okay, so now we've sort of closed the loop on all those membrane-bound proteins that we talked about way back in like September, and all those transport proteins and things like that. That's how they get there. Okay, so now we have that full story. Other proteins are going to get secreted from the cell, like mucin, like chondroitin sulfate, like insulin, okay, or whatever other favorite secreted protein that you like. And there are two major mechanisms for that, or two types of secretion, if you will. The first type is called constitutive secretion. And actually these two examples here, mucin and chondroitin sulfate, are good examples of that. So constitutive means it's happening all the time. Okay. And usually in those same vesicles are proteins that are inserted into the membrane that need to be, need to get to the cell membrane. So that's usually part of constitutive secretion as well. There are exceptions, of course, but generally speaking. So something that the cell is making all the time and secreting all the time. So these goblet cells especially are always secreting mucus because the stomach acid is always breaking it down, so you always have to replace it. That's part of this constitutive secretion. Insulin, though, is a good example of the other type called regulated secretion. We're not always dumping insulin into our bloodstream. When's the only time we need insulin? Right, you just had a big candy bar or a big donut or something like that when you have a lot of sugar. So the sugar, through a signaling mechanism that we'll study in another week or two, tells this, specifically your pancreas cells, that hey, here's a bunch of sugar coming your way, secrete some insulin to tell the rest of the body that it's coming along. So things like insulin that are only secreted under certain circumstances is what's called regulated secretion. And here's another version of that. So regulated secretion. Oh, constitutive secretion is sometimes also referred to as bulk flow. Is another term you might come across, but it's the same, sort of the same idea. So if there's no regulation to it, then sort of the default setting then is constitutive secretion or bulk flow. Okay? So anything without some additional signal gets put into those kinds of vesicles, hook up with the membrane. If you're in the membrane, you're still in the membrane, and if you're inside the vesicle, now you're secreted to the outside of the cell. Those proteins that are part of regulated secretion have other kinds of signals that sort of tell this vesicle, you don't fuse with the membrane until we get the right signal kind of idea. So again, insulin is a great example of that. Okay, so we sort of two types of secretion. And then the third major destination for things coming off the trans-Golgi are lysosomal enzymes being put into vesicles called lysosomes. And naturally, that's going to have its own separate kind of signal, separate from the regulated secretion kind of idea, because lysosomal stuff is not secreted, but it needs to be sort of gathered up and put into that little package, that little vesicle that we call a lysosome. And that's going to be the focus of our talk on Wednesday. So that will also complete the endomembrane system. Okay? So if you have no signal, you're part of bulk flow or constitutive secretion. If you have a special regulatory kind of signal, you kind of hang out until you're needed, and then we'll see what the special signal is for lysosomal enzymes on Wednesday. How are we feeling about that? Make sense? And so post-translational modifications of proteins and sorting of proteins, especially in the trans. That's really the job of the trans-Golgi especially. Okay, so stop it 2 minutes early today. Okay, we'll finish this up with lysosomes on Wednesday. That will complete